Good. I will start. Okay, so how to square microbial life in solar salterns. This is a small introduction to hypersaline life, life at high salt concentrations. But first, let us go a little bit to what really is salt, what is really hypersaline. And I think a good example is what I got when I was invited to go to a lake on the island where I work on Texel. And the newspapers briefed about this lake that turned completely pink and was hypersaline, according to the local ecologist. But when I went there, we measured the salt concentration. It was about 40 grams per liter, which is a little bit more than seawater. So yes, it's hypersaline, but only from the perspective of a aquatic freshwater microbiologist. For me, that's absolutely not hypersaline. So what I do work on more is like the salt lakes in Gironde in France, where you have the solar salter system, where salt is being harvested for commercial and industrial use, and you have kind of a microbial mats growing in these very shallow salt lakes, and you have all these fancy ring structures called fairy rings. So there you have about 150 to 200 grams salt per liter. But even more salt is my main topic would be the salt lakes, the solar salterns, and the crystallizer ponds that you find in Santa Pola. I've also done some work on deep sea brines, and you find also these very high concentrations in the brines that the astronomers think are present on the Jupiter moon of Ganymede. So these salt lakes are mostly commercial, but you also have some natural salt lakes. They have this beautiful coloration from algae, the cyanobacteria, green algae, and then at one moment the lakes start to turn more orange, more pink, and that is because of a lot of organism that has a lot of beta carotene accumulated, mostly as a protection against the high sun irradiance and the UV irradiance. Now, on this picture from Terry McGinnitic, you can see very clearly the kind of the holy trinity. Here we have three domains of life. We have the eukaryotic algae, we have the bacteria like Salinibacter, but we also have this very weird looking square organism, which turned out to be an archaeon. Now, this square was discovered already in the late 1970s, published in 1980 by Tony Walsbyi. At that time, for a long time, mentioned named Square Bacterium because they didn't know yet whether it was an archaeon. That was still a developing issue, and it has a very peculiar shape. It's like a post stamp. It's regular angles, uh, but it's flat. It's absolutely flat. It's membrane on membrane. One of the problems in further characterizing this was that it's unculturable. For a long time, at least until I came along, it was unculturable. And what is even more frustrating is that it's also the most abundant species in these brines. So you have a sample with 50% of these organisms are these squares. Go to the lab, you try to isolate, and you isolate everything except the square. Everything grows well in the lab except the square. So in the end, it took me about 2 years. So this is the moment where we started sampling the brines. You can see here is the remnant of the salt that's just been extracted for chemical use. And here, this is my friend and colleague Paco Rodriguez Valera from the University Miguel Hernandez in Alicante. And here we sample a small puddle of the hypersaline brine, almost a bitter with very high concentrations of sodium chloride and magnesium chloride. And it took me about 2 years growing on very low nutrient concentrations, low carbon concentrations. And finally, I had this organism into pure culture. Surprisingly, there was also a second isolate, was more or less in the same period, which was isolated from a saltern in Spain. I know in Australia. So uh, you can see here these nice electron microscopy images that they really have perfectly square form. Uh, all these white dots that you see are gas vesicles. They are filled with with air, whatever gas is around them, and it helps them to float around in in the brines and preferably be close to the surface because they require oxygen. They are aerobic archaea. I named it Haloquadratum walsbyi. It was well published already in 2004. So we also started to sequence the organism, and well, what could have been done now easily for about 100 euros, we had to do in the beginning of 2004, 2003, for over 100,000 euros. You can't imagine it anymore. This was the cost to sequence about 3 million base pairs and a small plasmid of 30,000 base pairs. But at least we had the genome, published it well, and we can dive further into the ecology of the organism. Now, I have different interests in my long career, and one of them are the daily and seasonal rhythms. And this is an example of a rhythmic movement of sulfur oxidizing bacteria in a coastal microbial mat. So low salt concentration. This is a stop motion movie. And what you see is the movement up and down with the availability of oxygen provided by the cyanobacteria. Cyanobacteria have a true clock. They have a circadian clock. They do photosynthesis during the day, producing oxygen. But at night, they are in the dark, they start fixing nitrogen. And that is a process that cannot tolerate oxygen. So this is why they separate these things. Very interesting, clear example of a rhythm in these examples, or in these samples. Now, when we go back to these fairy rings growing in Gironde at 150 gram salts, so this is a very shallow pond. There's only about 5 to 10%, 10 centimeters of brine above these microbial moss. They're not dry; they're in the water. And I hope I can show. So they grow very slowly, but every day a new ring is formed. If I shield it from the sun, there's no ring being formed. So it's a the photosynthesis depending process. The layers are like a mucus layer, and it's full of dead diatom casing. We do not yet know clearly what the process here is, but very interesting. So the question is there any rhythms in Haloquadratum, other uh, halophilic uh, archaea? And the reason is because Haloquadratum is a phototrophic organism. Doesn't do photosynthesis, but has a bacterial rhodopsin that produces, upon the incidence of light, captures photons, and the chromophore changes conformation, and protons are being pumped over the membrane. And this proton gradient can then be used to generate ATP via an ATPase. So it would be logical to spend as minimal as possible effort and energy, nutrients into making this protein. So, and in doing that, if you would expect that the gene is only transcribed closely before when it's needed. So you would expect kind of a circadian pattern in gene transcription to make this to produce this protein, and maybe also other proteins. So we did the transcriptome analysis with a cultured organism, and we compared the dark transcriptome with a night transcriptome, and unfortunately there was no difference. In the end, yeah, a negative result is also a positive result. These are the facts. There's no differences in our culture in the expression of any gene between light and dark. So, but we also were interested in seasonal differences. So we took samples from a winter and a summer from the same brines from which the Haloquadratum was originally isolated, but a couple of years later. And we again did a differential gene expression comparison. But there we saw a clear overexpression of a number of genes, including the bacterial rhodopsin during winter relative to the summer. And most of these genes are involved in the energy maintenance. So during the winter, there's less sun, lower temperatures, and the species Haloquadratum puts in more effort to compensate for this energy low period of time. What we also see, for example, is if we compare the gene expression patterns, these are the transcripts per million, and this is how many genes. You see a kind of a bell-shaped curve for the two natural transcriptomes from a natural ecosystem. But in comparison, the cultured strain shows a completely different pattern of gene expression. So most of the genes are actually low expressed, and only a few genes are very high expressed. And in nature, we see this bell-shaped curve with more average of gene expression patterns. And we think that is caused by the fact that our culture is only one species, but in the natural environment, you have many different ecotypes of the same species. Now, we came be aware of these ecotypes by doing this kind of a read mapping of a metagenome to the sequenced genome. And then we see this kind of these genomic islands, regions that are not well covered by the reads from the environment. And these regions are therefore unique to the sequenced genome. And that led us to the idea that, okay, there are probably different ecotypes. They are still the same species, but they have a different genetic makeup. Now, one thing has to be said is that, okay, the samples were taken about 7 years after the sample that I took for isolating Haloquadratum, this strain HBSQ001. So I went back. I didn't went back. I went back to the freezer. The fact was that what I did, I wasn't planning to isolate Haloquadratum because actually I didn't even know about its existence at that moment. But we took samples for for DNA extraction and doing metagenome analysis. But the isolation became a success, and the DNA stayed in the freezer for a long time. And in 2023, 2020, beginning 2024, I gained a lot of money finally for sequencing these samples. And so we started doing the sequence, but we find more or less the same. We find still these regions which are very low covered by sequencing reads. Still means that the original strain is less than 2% of the total ecotype diversity at the moment of isolation. So we can wonder, uh, did I isolate this species because it evolved in the lab during this 2-year period, or were these genetic differences maybe key for being able to isolate it? So one of the reasons why Haloquadratum is successful at these high salt concentrations is probably the presence of this very large halomucin, a strain that Nicole just also, or the protein that Nicole also just mentioned. It was at that time the largest archaea protein, more than 27,000 nucleotides, 9,000 amino acids. And it has all these kind of repeat sequences and a signal sequence, which is typical for mucins that we also have in our throat, our eyes, and so to keep our tissues moist. Also coming back to the idea of Nicole, this is very long gene, has been horizontally transferred. We do not know from which organism. It has a high GC, and there are these remnants of very low 80 or very high 80 around it, but it has all the characteristics of a horizontally transferred gene. Now, we showed that it was really extracellular, that it's outside of the cell. So it's a very large protein, and it has to be transported through the membrane, which is also a very energy costly process. So we predict that these mucins, like we can see in this picture, this is the side of Haloquadratum. So you look at the flat side. There you see this mucin being formed, but it's a protection against desiccation and potentially also a protection against viruses. So in conclusion, hypersaline ecosystems are very rich, very biodiversity rich. They contain each domain of life, including the viruses. They are very important for understanding cellular evolution and model ecosystem for astrobiology, which is interesting again for the work of Carlos. And so maybe potential questions for the panel later is, could they also be tell us how life evolved, and especially do they have a potential for extraterrestrial life or for maybe terraformation, etc., etc. And with that, I thank you.